Tiempo. Ya estamos en el. ¿Tú? 5 minutos. Digas. ¿Sí? No, sí, como 5 minutos. Ok, 5 minutitos nada más.
Ah, no, es que es una de.
¿Tienes formado? Claro, pues sí, viste. Ay, sí. Ya ves, vámonos. Que aparte de llegar y no había nadie. Ay, sí, hasta da miedo, parece justo. Pues, impresión muy militarística. No, bueno, sí, sí, no, gracias.
Firme.
I'm going to introduce you our head of the department, Doctora Julieta Garduña.
Thank you.
Nice to be here.
Nice to meet you.
Head of department.
Thanks for coming. Yeah, she is all of us. So I love Mexico. This is my first time in Mexico City.
Oh, okay.
But I spent some time sightseeing yesterday afternoon, and I'm going to take all of Saturday.
Okay.
Okay.
And I love it here.
Enjoy it.
How long will you be here?
I'm leaving on Sunday,
Okay.
but I deliberately added in an extra day after 2 with Pascal, so that I can just roam around. It's a big city though.
Yeah.
Boston, 1 million, the whole metropolitan area.
Yeah.
And so my plan to roam around on foot maybe won't work out so well.
Yeah, it's really
It's really near.
Mhm.
Yeah.
So it's a really beautiful place.
Yeah.
But-
Very, very
Yeah.
beautiful.
Oh.
Yeah, you Federico. Pues sí. Vamos a ver, fue muy de cómo va apresurado.
Qué bien.
Pues vamos.
Sí, listo.
Whenever you guys are ready.
Bien, buenos días a todos y a todos. Muchas gracias por estar aquí con nosotros el día de hoy. Para mí es un gusto, ya saben, presentar estos ya tradicionales seminarios del Departamento de Psicología. Este en particular es un seminario extraordinario y quien nos acompaña el día de hoy es el doctor Donald Katz, quien, bueno, cabe mencionar que fue invitado por uno de nuestros investigadores del departamento, que fue el doctor Jean-Pascal Morin, y pues quien nos hará el favor de hacer la introducción del seminario extraordinario del día de hoy. Dr. Katz, thank you to be with us today.
Thank you.
And adelante, doctor.
Bien. Muchísimas gracias, Ricardo. Muchísimas gracias a todos por estar con nosotros el día de hoy. Voy a pasar al inglés, si me permiten, y también me perdonan mi acento. Muchos sospechan que no tengo acento, pero está muy bárbaro. Introduction. I'll give it later. I've been knowing Dr. Katz from the papers since I was a PhD student, so not so long ago when I was. So yeah, I was with Federico, as you all know, and that was he wrote some of the challenging papers, and one of the first ones that I introduced to my new student when I got with Gabrielle in the lab. Fortunately, they got to meet Donald in one of the Society for Neuroscience meeting, and yeah, well, we stick together, and we have the intention to invite him here and collaborate and everything. So thank you very much for coming here. So I give you a brief resume or semblanza of Dr. Katz. Dr. Katz underwent undergraduate training at Brown University, after which he completed his PhD in psychology and neuroscience in Indiana, at University of Indiana, where he trained in vivo electrophysiology. So, like all of the stuff you're going to see today, pay close attention in vivo, like behaving rats in vivo electrophysiology. Uh, so he next worked with Miguel Nicolelis and Sidney Simons, big name in taste perception and sensory motor function. So during his work, he provided evidence that the taste perception is multimodal, multimodal. So it's also hedonic part, the sensory part, are many parts. It's a very rich experience, and it's also distributed. So encoded by distributed and many brain areas, amygdala and insular cortex, whereas the gustatory cortex. Now he runs the CATS lab at Brandeis University. He's a full professor in the Department of Psychology at this university. He's been recipient of multiple academic honors, including the Pollack Young Investigator Award from the Association of Chemoreception Sciences. And he also had the Janet Lerman Neubauer and Joseph Neubauer Prize for Excellence in Teaching. So without further introduction, I leave you with Donald.
Thank you. Thank you, Pascal. So I have a very scary, first of all, thank you very much for inviting me to come here. This is my first time in Mexico City. And I must say it's overwhelming, and I'm having a wonderful time. But I love cities, and I'm looking forward to having a chance to both talk to more of the students and also get out and see more of the city. I'm going to present a talk this morning with a very scary title. I understand that this is not a program in which a lot of this kind of work gets done. I promise you that the talk is not as scary as the title, but please do not be afraid to interrupt me with questions as I go, because if there's something you don't understand, two things are true. One is the talk will get worse and worse as it goes along, and the other is that all of the people around you probably don't understand either, and it's my fault. And so you become the hero if you speak up. And so please don't let me get away with not being clear. And so what we're really talking about here is, in fact, very simple. The reason I study taste as opposed to some other sensory system is because while you can show an animal, be it a human or a non-human animal, a picture and expect that that animal will have no particular response, the neurons in the brain might respond, but the picture means nothing. While you can present an animal with a sound, a sine wave tone, boop, and have that animal just shrug it off, it is completely meaningless to the animal. Tastes never are. If you put a taste in an animal's mouth, it is a meaningful stimulus about which a decision must be made. And that's why I study taste. If you put a taste in a rat's mouth, you can actually see the animal making a decision as to whether it likes it and wants to swallow it or dislikes it and wants to spit it out. And so here is a video taken by my very first graduate student, Steve Grossman. This is a rat sitting in a box, and you can see, although it's a little blurry, the business end of this rat, the front, and the mouth is right here, and there are the forepaws. What you can't see is that via an intraoral cannula, Steve is about to put a small drop of sugar water in this animal's mouth. And when it does, it produces these licks that essentially tell you that that is yum. That is the rat yum face. That is the specific face the rat makes, moving its tongue around from side to side inside the mouth when it's letting you know, I like this and I'm going to swallow. And on the other hand, take the same rat and put in its mouth a dollop of quinine, which is very, very bitter, although with a little sugar added, it is the second best ingredient in a gin and tonic. And if you put that in the animal's mouth, you get an entirely different reaction, which is called the gape, which is showing you that the animal is not happy with what's in its mouth. And there's one, and there's another. They come in little bursts. What you can see is the jaw opening wide, like in a yawn, the teeth protrude. What you can't see is that inside the mouth, the tongue is rolling forward to expel the fluid. And that is the rat yuck face. And so with that, even with an untrained rat, we can see the decision process as the rat takes this taste stimulus and decides what to do with it. And that with that, and I should mention that it's not just rats. This is a human infant. This was my human infant who just put a wedge of very sour lemon into his mouth, and he is making a classic gape. Now, people often ask me, what's the difference between a rat and a human? The answer is not so much. But rats are smarter than humans. The moment after I snapped this picture, he put the lemon back in his mouth. And at the entire other end of the phylogenetic spectrum, there are toads. These are toads in the Kalahari Desert. And you probably think of toads as being very innocuous creatures. The most dangerous thing they might do is pee in your hand. But if you're out in the desert, they're hunters. And if you are a beetle, they're very dangerous. And so the beetles in the Kalahari have developed wonderful defense mechanisms. And the hero of our story has evolved a chemical on its shell that makes it taste like shit. And so watch what happens when the toad tries to put our beetle in his mouth. And that is a gape. And just to prove to you that toads are dumber than both rats and humans, wait for it. Oh, hello. The video has paused. Oh, well. If the video kept going, you could see that other toads also tried to swallow the beetle, also made the gapes. Our hero escapes. Gapes are ubiquitous across huge swaths of the phylogenetic tree. And so with that, we go and we do electrophysiology. The way we do it is we actually take bundles of electrodes and in a surgery insert them into the brain of the rat, and we cement them in place. And they are outside the cells. These are extracellular recordings looking at action potentials extracellularly. Please, if these are terms that any of you are unfamiliar with, please let me know. And we can record from the activity of multiple neurons simultaneously as the rat tries to consume these fluids. And what I will show in this talk on the basis of these data is, first of all, that time matters in neural taste processing. If you have read anything about taste in the brain, you've probably read that it's as simple as a neuron goes on for sweet, a different neuron goes on for bitter. I'm happy to talk about this afterwards. Let me not mince words. That's just wrong. Time matters. And through watching these responses, you can actually see the processing occur in the animal's brain by watching the neural activity. And so you've got action potentials occurring, and you've got action potentials occurring in a particular pattern, say 2 quick and then 1 after. And it turns out that just talking about those in terms of, oh, that's 3 action potentials, does not tell you what the process is, because the process has to be looked at through time. Another thing we're going to talk about, in addition to the fact that time matters, and I'm going to show you that cortical taste responses evolve across a second to a second and a half, and that this evolution of response is meaningful. I'm also going to tell you that neural ensembles, so groups of neurons, work together in the processing of taste. Doesn't make sense to look at each neuron separately. They're connected to one another. The process is an ensemble process. And that means that single trial dynamics, the response through time within a single tasting, is coherent across ensembles, and it's nonlinear, and I'll explain how. And the coherent nonlinears drive behavior. And I'm going to show you some fancy math, but then I'm going to show you how what you see can be simply explained in terms of what the animal is doing. And finally, it's not just that neural ensembles work together to process taste, so do brain regions. And I'm going to specifically focus on the amygdala, which I know is one of the favorites of some of the people in the room. The amygdala, as it turns out, and this we're going to delve into some work that has not yet been published, showing that the amygdala couples with the cortex into a single processing unit. And so talking about amygdala and cortex is actually the inappropriate way to do it, because the amygdala cortical complex is working together, enabling these nonlinear dynamics. And that's the whole talk. And so what I'm going to do now is introduce you to the electrophysiology. This is always an embarrassing moment for me. What I'm going to do now is take my 5-year postdoc and boil it down to one slide. When I was at Duke working with Nicolelis and Simon, what we found was essentially this. When you record from a single neuron in gustatory cortex, the primary taste cortex, and you deliver a range of tastes one at a time, and you do PSTHs, peri-stimulus time histograms, so that's the firing rate of this neuron across time after the stimulus arrives. And now I've just piled the different responses on top of one another so that each color is the response to a different taste in this neuron. You can see that it's not as simple as the neuron goes on, the neuron goes on. And in fact, after a great deal of analysis that I'm not going to take you through, because I got a lot of other analysis I want to take you through, can tell you that this response falls into 3 parts. And you can even see that just from looking at it. The initial response is a burst of action potentials, and that burst of action potentials is devoid of information about taste. It's pretty much the same response to all taste. And so we in the lab call that the detection response, because looking at that, we, the investigators, can detect that a taste has hit the tongue. We can't tell what the taste is. I put it that way very specifically because I don't want to say that the rat is using that response to detect. We don't know that for sure. And then after that, a few hundred milliseconds after that, you can see that the responses to the different tastes become different from one another. And so in this period, we call this the identification epoch, because looking at the different responses, we can tell as the experimenters what taste was delivered. We can answer the question, what is it? And I can tell you that each taste neuron has a different pattern of response during this period. There's a wide range of different responses that we can get, but with just a few of them and very simple analyses, we can say, okay, I now know what taste it was the animal was tasting. But then you can see that the responses change again. And when you get out to this portion of the response, which emerges between 0.5 seconds and 1 second, now in this neuron, you'll notice that the response to quinine, which is the nastiest taste, is the highest. And the response to sucrose, which is the most delicious taste, is the lowest. This neuron is now responding in a palatability-related way, which is to say, the ranking of its responses matches in reverse the ranking of how good the tastes are. And I should mention, about half of the taste neurons do this, about half of the taste neurons do the opposite. But we can do, again, simple analysis. We do a correlation, a linear correlation between the known palatabilities measured in terms of how much the animal will consume and the firing rate of the response. And you can see that as you get into this late palatability period, that correlation becomes significant. And so in this late epoch, now the neurons are responding in a way that is significantly related to the palatability. And we as experimenters could answer the question, should the rat eat the taste? And what I'm going to show you with the most of the talk is that we know that that's what the rat is doing as well. And so that is the basics of the taste dynamics. Yes?
Adaptation, no?
Mm-hmm.
It has to do with the receptors, or even when we teach the sense, no?
Mm-hmm.
And it has to do with that, no? Because the bitter of the taste is, I correlate with pain.
Mm-hmm.
That you never will adapt in sensation.
Right. So certainly there are a lot of processes going on, and one of the things I'm not going to talk about in this talk is that we look at different timescales, and it turns out that if you deliver the same taste several times in a row, the response changes, and we see all sorts of adaptation responses. We see responses also that change with psychological variables like how bored the animal is with the session. But within a single trial, we're pretty sure that this is not an adaptation mechanism. And certainly the earlier evidence suggests that across a single second timescale, the bitter receptors are still active. That if you deliver one dollop of taste, one aliquot of taste to the tongue, the bitter receptors will fire like gangbusters for a second, 2 seconds, 3 seconds. It actually takes much more than we're delivering to cause that sort of adaptation. And then we deliver water to clean the tongue. And so we are working very hard to avoid that sort of adaptation response, which is not to say that it doesn't occur. It is to say that it doesn't affect this. So, okay. So just want to do a little side note, just because one of my senior graduate students is doing work right now that is still in prep. And I'm just so excited about this work that I wanted to present it to you. Most recently, she has shown not just that these neurons culminate in firing related to palatability, these neurons culminate in firing related to the particular animal's preference. And so when you go through, it turns out that different animals have different preferences. Here's 4 different rats, and the difference between they're licking to sucrose and sodium chloride. And you can see that for 2 of these rats, they strongly prefer sucrose. 1 of these rats prefers sodium chloride. 1 of these rats doesn't really prefer either of them. We can do the same thing even with quinine, which is bitter, and citric acid, which is sour. And that when we then go and look at those animals' taste responses, the correlation with palatability is higher when you correlate it with the individual's taste preferences than when you correlate it with the canonical taste preferences, which is what we've done. And so these late responses are reflecting what that particular animal likes and dislikes. But this is all single neuron stuff. We're recording multiple neurons at a time. And one of the basic things we got wrong in looking at stuff like this is that this makes it look like the palatability correlation comes on slowly. This is across from 500 milliseconds to a little bit over a second. That's infinite amount of time in the brain. Half a second is a long time in the brain. And we had thought that that was what was going on, that it was taking a while for this to emerge. It turns out we were wrong about that. And let me show you how we figured that out. When you record, instead of doing a PSDH, in which you take a bunch of trials of the taste and you sum an average across them, you take a single trial and you look at a bunch of simultaneously recorded neurons, you see something very different. And so this is a recording done by my first postdoc, Alfredo Fontanini, and analyzed by my second postdoc, Lauren Jones. These are 10 simultaneously recorded neurons. Each of these hash marks is an action potential fired by that individual neuron, and this is a single trial of quinine. And while it's hard to see anything in this, which is why these sorts of analyses are so seldom done, I'm going to give you a little help and say if you look right at that moment in time, which is at about the time when palatability coding is at its peak, you'll see that a sudden change occurred in firing. And let me help you. This neuron, this neuron, this neuron, this neuron, and this neuron all suddenly start firing at that time. This neuron and this neuron all suddenly stopped firing at that time. It's there. It's very clear once you're shown it. This is what happens in single trials, is that there isn't a gradual process. Around this time, there is a sudden ensemble transition such that using an analysis, a machine learning analysis called hidden Markov modeling, which, by the way, for those of you who are interested, is the analysis is what you use whenever you are talking to a machine on the phone. When it asks you, when you call the airline and you're talking to the computer and it asks you for input, you are talking to a hidden Markov model, the job of which is to divide up these multiple time series into parts. And using that, we can detect that there is a state prior to that change in which neurons are firing at one set of rates and a state afterward. Furthermore, we can talk about the period of uncertainty, which is really, really small during the change. We can even use the analysis to say, what is our confidence that the set of neurons is in one state or the other? This is all fancy math, but it basically allows us to show that this is a very sudden change. And that sudden change, you might ask, okay, well, you put up one thing that showed slow change, you put up another thing that showed fast change. How does it put together? Here are 10 consecutive trials of a single ensemble of neurons. And what I'm showing here is this plot of our calculated confidence in the onset of the state that is dominant out here during the palatability period. And you can see that in each trial, that state comes on suddenly, but in each trial, it comes on at a different time. And when you average those together, you get a slow ramp. And so this slow ramp in palatability was an artifact of us analyzing the data incorrectly. And that when you look in single trials, that comes on quickly. Now, mind you, what I haven't shown you up to this point is that what's really happening is palatability responding is suddenly coming on. And so what I have to do now is I have to show you that the state before this is not palatability related, and the state after the transition is. And so this was the work of my graduate student, Brian Sadaka, who in 2016, whole separate data set, basically started by doing the same thing that we've always done to show that we're still getting it. And I should tell you, after 20 years in this, doing this, every time a data set comes back showing this pattern, I still go, whew, because you're never quite sure that you're going to replicate. And so, but when he looks at his sets of neurons, he sees what we've already seen, correlation with palatability, starting at about 0.5, going on to about after 1, increases. And so then he did hidden Markov modeling on every one of those trials. And he basically said, okay, there's where palatability is high. So we're going to say that this cyan colored state is the putative palatability related state. And then he did a very simple move. He said, this is when the state comes on to full in each trial. And then he pushed the trials around so they were no longer synchronized to the onset of the stimulus. They were synchronized to the onset of that state. And then he just took that slightly changed data set. And by the way, we're talking most trials, there was a shift of 150 or so milliseconds. Some trials, it was quite a bit more. But when he did that shifting and then redid this basic analysis of how palatability correlated it was at each point in time, he saw a dramatic increase in suddenness. In fact, now I'm going to not go into all this analysis. I'm happy to talk to you about it afterwards or point you to this paper. We did a whole bunch of analysis, and we can tell you that as far as we could measure, given the vagaries of the analysis and of neural firing, this transition into palatability responsiveness is effectively instantaneous. One moment it's not there, the next moment it's there. The way I describe it in talks is when you're watching television and an actor comes on the screen and you say, I know who that actor is, and suddenly it comes to you. It doesn't come to you slowly. It's Robert somebody. It comes to you all of a sudden. The next trial, the next actor, it will also come to you suddenly, maybe slower, maybe faster. That's what's happening here. But this is all still phenomenology. I've shown you the activity and shown you what it's related to. I would say it's pretty compelling, but I want to make it more compelling because I want to drive home the point that this really is what's happening. The animal really is going, oh, I recognize now that I don't want that taste in my mouth. So the first thing we did was we took a whole bunch of trials, and because we can look at those gapes and know when those gapes go on, we can have the latency of the animal's decision to reject quinine. And we can also look at, in those same trials at the same time, the latency of that transition into that late state. And we can relate those two. And if all of the data points for all the trials fall on this unity line, it means the two happen at the same time. If the data points fall up here, it means the neural transition happens and then the behavior happens. If the data points end up here, it means the behavior happens first. And here are the data. With the exception of 4 lonely trials in this entire session, it's not just that in each trial, the behavior follows close after the transition. It does so despite the huge variation from trial to trial in latency. And so sometimes that decision happens early, sometimes that decision happens late. Always Gaping happens right after, and when I mean right after, the average is about 300 milliseconds from that transition. And so we'd say that this is pretty powerful evidence that that's what's going on. But let's do some manipulation. First, I'm going to do the psychology manipulation because we're actually psychologists. And the first thing that occurred to us to do was to try to manipulate the system through cueing. And so what my postdoc Jen Lee did was she basically delivered, she cut down the set of tastes she was delivering, so to just sucrose and quinine, and she delivered strong concentrations and dilute concentrations. You don't have to worry about any of that. All you have to know is that the only taste to which the animal gapes is strong quinine. And so she basically did one extra thing. Every time, just before she delivered strong quinine, she played a tone. And so through the session, a smart rat will learn when that tone is coming, oh no, something I hate is coming. And when an animal gets nervous like that, it decreases the latencies. I was talking with Pascal. If you play that game where you're holding your hands like this, someone else is holding their hands like this, and you know that they're going to go slap, it decreases the latency to that move, sometimes making it happen even before they move. So we expected that this cueing would decrease the latency of gaping, and in fact, it did. And just for comparisons, she did sessions in which she didn't cue, and from the first half of the session to the last half of the session, there was no change in gaping latency. But she was recording from the neurons at the same time, and in these sessions, across those two halves, that transition also decreased by exactly the same amount. And so when we manipulate how fast the animal gapes, we are also manipulating how early that transition occurs. But amongst biologists, what they really want to hear is what happens when you screw with the brain. And so, as with all things, my lab refuses to do things the normal way. And so I'm now presenting the work of my graduate student, Narendra Mukherjee. And what he decided to do was use optogenetic silencing to actually manipulate the activity of gustatory cortex during one of these trials, but we did it in an unusual way. Here, by the way, is a picture of just a slice. There's gustatory cortex down there, and you can see from this light that he had infected this area with a virus that codes for both green fluorescent protein and also ARCH-T, which is an optical silencer. And then he put fiber optics in here in a second surgery so that he could light those neurons up and thereby stop their firing. And he decided to do it, though, in each session, he decided we were going to do 4 different trial types. On 1/4 of the trials, he basically turned on the light for half a second at the beginning of the trial, just half a second. And I should say arrogantly and smugly that most researchers, when they use this sort of technique, they just turn on that laser and they blast the hell out of the brain for seconds or minutes at a time. And we delivered 1/2 second of inhibition. And by the way, we've got data when you turn on that laser for more than a couple of seconds, you heat up the brain. And so on another 1/4 of the trials, he delivered the laser for this half second. Another 1/4 of the trials delivered the laser for these half second, and for 1/4 of the trials, no laser at all. So all of the analyses here are within animal. They're all within session. And keep in mind that the range of transition times, the range from trial to trial in behavior and transition times can be as early as 0.5, can be early as 1.5, which means that the transition, this particular middle trial type will sometimes happen before the transition and sometimes happen after the transition. And that's what I want to focus on, because I can tell you all about this data set. There's a lot of cool stuff in that data set. But let's just focus on the fact that the trials in which Narendra turned the laser on at 0.7 seconds and turned it off half a second later. In this data set, in Narendra's hands, gaping happened on average at about 900 milliseconds in. Keep in mind, and this is something that I'm going to talk, that I'm happy to talk at length about, when the animal gapes is dependent on a lot of different things, including how scared the animal is, including how strong the quinine is, including all sorts of different things. In this particular data set was about 900 milliseconds, which means that it happened on average during the time that that laser was on. And so, but again, that range of times that the transition could happen is all the way across here. And so that means that there were some trials in which the laser went on before that transition happened. And Narendra was also recording, so he could say, okay, that neural transition hadn't happened yet. And while the average time of latency to gaping was 900 milliseconds, on those trials, the animal didn't gape for more than 600 milliseconds. The animal could not gape until the laser went off. And so that is strong evidence that that this transition is in fact related to gaping. Even better though, is in the same trial type, there were some trials in which the transition happened to happen early and happened before the laser went on. And in those trials, he had no impact. This looks earlier than 900 milliseconds. That's just because those were the earliest trials. And so on average, those gapes happened early. The upshot is, if he missed the transition, even by 25 milliseconds, it didn't have an impact. You have to block this transition to block gaping. And so, things to note before I move on to the last part of the talk. When you're asked the question, what happens when you inhibit GC, gustatory cortex, what these data show you is that the answer is, it depends. And one of the soapboxes, one of the preaching that I'm happy to do is that in neuroscience, there is this general belief that if you knock a bunch of neurons out, you can figure out what they do. You can't, because what they do depends on when you do it. And it depends even more subtly on what those neurons happen to be doing just before you turn the laser on. And so the question, what does a set of neurons do, is not answered by this kind of experiment, unless you put a couple of asterisks after it. Another thing is that gaping does eventually happen. These animal does gape. Yes, in general, it had to wait until the laser went on, but it isn't like we derailed the process entirely. The animal does end up gaping. And that means that some other brain area has to be involved. We knocked the hell out of the GC neurons, and yet once we let them free, gaping occurred, which suggests very strongly that there are other parts of the brain that are involved in this process that are keeping it going. And so that's what leads us to the basolateral amygdala. And so why the basolateral amygdala? First of all, we love it because it's reciprocally connected to GC. So BLA and GC talk to one another constantly. Second, it is responsive to tastes and on the same clock as GC. Here is a single neuron in BLA, its response, and we've noted those original epochs, and you can see that there's a change at this first epoch, and then a change here, just like in gustatory cortex, with one big difference. GC, that first epoch we called detection, the second epoch we called identity, and the third epoch we called palatability. And by the way, I'm numbering now the transitions between them because that's going to come into play in the last bit of my talk. If you look closely at the BLA response, you can see that in this middle epoch, this is a palatability response. This response to sodium chloride and sucrose is low. The response to citric acid and quinine is high. This is a neuron that responds more to nasty taste than nice taste. Once again, half of the BLA neurons showed us the opposite. But the point is that while the gustatory cortex is still reporting identity of the taste, the basolateral amygdala is already reporting its palatability, which made us think, okay, maybe what's happening at this transition point is that this information is getting passed to cortex. And I know there's a lot of questions as to, well, how would that happen? I'm not going to answer those questions today, but I'm going to get us in the direction of that. And so these things led us to think that the basolateral amygdala is probably very important for this. And finally, also, the BLA is well known to be involved in driving emotion-laden decisions in learning. This goes way back to the human work of Antonio Damasio and the rat work of a large number of people, notably Jeff Schoenbaum, who I list both because he's one of the smartest of them and because he's a personal friend. And so in both humans and rodents, it's been shown at time and time again that BLA is involved in this. And in fact, Damasio, early on and on the basis of human work, suggested something that looks exactly like this. He suggested that BLA processes the emotion and then passes it to cortex, which acts on it. Okay, so now we're going to look into this BLA to GC connection, and this now is the work of one of my postdocs, Zhenyu Lin. It must be obvious already that in my talks, unlike some researchers who like to present a paper in great detail, I like to shout out to everybody in my lab. And so this is the work of Jean-Yu Lin, and what he did to make the first look at the relationship between BLA and GC is he did an experiment in which he infected BLA with ARCH-T. Okay, now all the neurons in BLA are expressing this optical silencer. The problem is, that's all the neurons. They project all over the place. But what he did, and to be honest, we weren't sure this was going to work, is he then put his fiber optics, the ARCH-T infected anterogradely, and so the axons of these BLA neurons were infected, and he put his optotrode, which are electrodes and a fiber optics, into GC, so that when he lit up that laser, the only thing he was knocking out were action potentials coming from BLA into GC. And so he did that experiment, and what he found, and mind you, this was, he was already doing a complex enough stuff. We did not replicate Narendra's multiple trial types per session thing. Instead, he just turned the laser on for a second and a half. And what he discovered is that when he did that, he massively decreased the amount of palatability relatedness in those GC responses. And so knocking out the input from BLA removed almost half, removed the palatability responses in almost half of his neurons. Now, mind you, and this is worth noting, it actually caused palatability responses in a small percentage of neurons. And so something fancier than just BLA communicating all palatability GC is going on. We'll get into that another time. The big result there is that mostly what knocking out the input from BLA does is reduce the palatability coding in cortex. And by the way, this is also confirmation of a much earlier paper from my lab in which we did a similar thing using pharmacology. But much more interesting to us is that this was not the most impressive result. This was not really what we found was the most compelling thing that knocking out BLA input did. What knocking out BLA input to GC did was it changed the dynamics. We're looking at GC. Here is the intact animal, and this is essentially looking back at what Brian Sadaka did. This is showing the precipitous onset of palatability-related responding in normal animals, comes on very fast. And when on trials in which he knocked out the BLA input, that precipitousness was gone. The slope of that transition was reduced by more than half. And so knocking out BLA's input to GC, the biggest impact it has is it screws with the dynamic process, which we've already shown you is related to behavior. Moving forward, and now what we're doing right now, what Jean-Yu is doing right now, is the experiment that hopefully is on a lot of your minds. He's now looking at how that messes with behavior. We haven't done that yet. Bring me back next year. But finally, one other thing I want to do, though, is to go into what happens when you record from both of them at the same time. And this is work from a graduate student in my lab, Abu Mahmoud, who is now staying on with me to do a postdoc for reasons that pass understanding. And he is not only a brilliant physiologist, but also a recovering physicist. And so this is the most computationally heavy stuff, and I've saved it for last. Because everything that I've shown you thus far suggests to us that BLA and GC must be working together to make this process happen. And so Abu went ahead and he actually implanted simultaneously in these two regions, recorded ensembles of neurons at the same time. And the first thing he noted was that those transitions that happen in GC that I've told you more about than you really want to know are also happening in BLA, and they're happening at the same time in individual trials. If the GC set of neurons transition early, so does BLA. If the GC transitions late, so does BLA. From trial to trial, the variability in when that transition occurs is something that's shared between the two regions. And in fact, it's specific for the transition in and out of this state. The significance of that coupling, the significance above chance levels, isn't in that first transition. It's only in the second and third. And so what's going on is these two regions don't start out coupled, but while they're processing the tastes, they snap into synchrony. And in fact, when I say snap into synchrony, the first transition, if you look at when it occurs, it generally happens in BLA first, which we didn't expect. It means that somehow taste information is getting to BLA before it gets to cortex. But the second and third transitions are synchronous. They are happening at the zero time point between the two. They are happening at essentially the same time. And so that brings us to the very last stuff I want to talk to you about, which is, okay, if these two are working together, how does that look? And so this is stuff that is also still in prep. And again, I can never leave this stuff alone. And if anything, I'd love commentary on the stuff that's still in preparation, because this is the stuff that you have a chance to actually fix. But so in looking at this, in looking at the question inspired by Damasio as to whether, while you're leading up to this synchronous transition, BLA is doing something to help GC. BLA is somehow passing stuff to GC. He used a very fancy analysis that I can unpack for you in the question and answer period if you'd like, called Granger spectral causality. When you talk about two things working together, typically you talk about them in correlation terms, in coherence terms, which is non-directional. It just tells you that two things are doing things in a correlated fashion. What Granger spectral causality does is it takes use of things like the lags between the regions to answer questions about what direction is the influence going. If these two are correlated, which one is driving which one? And of course, when you do this and you can look at both directions, it turns out that the answer is they're both driving one another, but not at the same time. And so here is the results of this analysis looking at BLA driving GC on what you see in this plot. This is weak influence, this is strong influence. So when you've got yellow, that means BLA is pushing GC. This is frequency range. Almost all the action happens at low frequencies, and you need to look at frequencies to understand these sorts of analyses, but I can unpack that for you. But the most important single thing is, look, BLA is influencing GC during one period and one period only. And that is that middle epoch, while amygdala is already coding palatability, leading up to the time that GC is coding palatability. BLA is, in fact, specifically influencing GC during that period. Furthermore, if you then look at the GC influence on BLA, you'll see there's much less of that. GC has basically taken it all in and then briefly has a strongly significant output of influence when, at the time that transition occurs and it's driving behavior. And so you can see in a sort of a space-age fashion, the correctness of some of these classic theorists, because what's going on in the cooperation between these two areas is that first BLA is talking to GC, telling it what it should think, and then GC goes, okay, then, gate. And one last piece of data, and then I'm going to stop. When you look at this, you can say, okay, well, there are probably going to be some neurons in GC that are actually connecting with BLA, some neurons in BLA that are projecting to GC. Can you tear it apart to look at what's actually going on in these different sets of neurons? And yes, we can. Abu used a much more classic kind of analysis, which is called general linear modeling, or GLM, to basically take individual neurons on the basis of the Granger spectral causality and say, which of those neurons are the ones influencing the other agent? And so what he found, for starters, is not very interesting. If you look at GC, understand that this is a relatively small percentage of the total number of GC neurons, but the number of GC neurons that are influenced by BLA are about the number of neurons that influence BLA. And there are a few neurons in the middle that are doing both. We did a whole bunch of analysis. The number that are doing both is exactly what you'd expect by chance, given how many are doing one. This is just this itself is not a very exciting conclusion, except that now with this, we can say which neurons are those and what are their taste responses looking like. And we can show you that in terms of both taste specificity of the middle epoch and in terms of palatability related responding of the late epoch, the neurons that are influenced by BLA are the ones doing all the work. And so the neurons in GC that are projecting to the BLA aren't actually the ones doing taste coding. Why is this? We don't know yet. I can also tell you that to make matters weirder, what Abu didn't give me to put up here is that the neurons from out here, the neurons that are neither sending nor receiving, are actually not doing any worse than this. In fact, there's a huge range. But the coolest piece of data is that when we just look at these neurons in the middle, when we look at the neurons that are both sending and receiving, those are where the meat really is. This, by the way, these plots, I don't know why Abu chose to do it this way. One of the things about having a bunch of people doing the work is that you'll notice that each time I present a different person's work, it's presented differently because that person did the analysis and the figures. What Abu chose to do here is this is the average, and this is the 75th percentile, and this is the extreme value. And so what you can see is that even though this is much higher than this, the difference between the means is actually smaller than the difference between that mean. What this says is that the more deeply embedded a neuron is in the circuit, the more it is involved in that back and forth, the more involved in taste processing it is. It really is the interplay between these two regions that's making all this happen. And that's all I have for you today. And hopefully I actually was successful in making the talk not as scary as the title, but in case it wasn't, let me give you my obligatory summary slide. First of all, GC taste responses, gustatory cortical taste responses reflect taste processing across the respective successive ethics. It isn't the case that one neuron turns on for sweet, another neuron turns on for salty. I refer to that, by the way, as the Downton Abbey theory of brain function, where, you know, the rich people upstairs, when they wanted the footman, would just ring a particular bell. There was the bell for the footman, and then there was the bell for the driver. And there seem to be people who actually believe that your brain works like that. It doesn't. It actually, you can see the processing. You can see into the brain of this rat as it basically goes from saying, okay, there's something on the tongue. Hey, it's quinine. Ew. Second, ensemble analysis of single trials reveals the onset of palatability coding to be a sudden ensemble transition. It isn't the case that the animal slowly says, you know what? I don't think I like it. The animal gets to a point and realizes, no, that's not what I want in my mouth. Just so you know, we are currently doing the same kind of analysis on the decision to consume, which is a lot harder, as it turns out, to detect in single trials, which is why it's slower coming, but it's pretty clear that the same thing happens there. This transition, in turn, drives behavior, probably by modulating a brainstem central pattern generator. And here, I just want to send a shout out to my boss and mentor, Eve Marder, who is the biggest of the big dogs at Brandeis. Love this person. She is one of the greatest neuroscientists in the world. If you don't know her work, leave here right now and go look it up. She has spent her career studying a set of 20-some neurons in the crustacean, in the lobster and crab. And this set of neurons is what drives consumption behavior in that beast. And it produces rhythms that cause the gastric mill to crunch. But what most of her career has been spent on is the descending influences from other aspects of the animal's brain and even the animal's humoral system that modulate what and when that rhythm is produced. And what we are arguing is that gustatory cortex is essentially working as a modulator on a brainstem CPG in much the same way. If I accomplish nothing else with my neuroscience career, I would like to rescue the term modulation from second-class status. That this is a very important thing to do. GC is basically telling a brainstem central pattern generator, go, time to give. And finally, the mechanism by which this transition is constructed involves the coupling between GC and BLA into a functional unit. We're pretty sure it's not just GC and BLA. I've got a senior graduate student working on lateral hypothalamus. I've got another one working on the brainstem central pattern generator itself. There's a lot going into this in addition to these regions, but these recent data make it pretty clear that you need this circuit to make that happen. And that's all that I've got. So, well, for the morning talk. And so I give you greetings from the Behavioral Learning of Electrophysiology of Chemosensation Lab. Yes, we are blah. There are a lot of people in my lab, and I don't really, I've introduced them as I've gone along, but I would like to shout out, these are the people who contributed to this work. We have postdocs and graduate students. The work I presented along here wouldn't have happened without all of these people. Also, I didn't underline her, but I'd like to point out my graduate student, Katie Kimbrell, is also an artist, and she made the cute little rat drawings at the beginning. Also like to thank, I have a lot of collaborators at Brandeis. Brandeis is a very collaboration-friendly place. And so these are my ongoing collaborations, but in particular, Paul Miller, who is also a recovering physicist, is the reason that all of this works out. To be completely honest, I can barely add. And so Paul is the one who makes sure that our analyses are rigorous and doing what they're supposed to do. Also like to thank funding from the National Institutes of Health, the National Science Foundation, and the Schwartz Foundation for Computational Neuroscience. I brought it in about 50 minutes, and I'd like to thank you all for your attention. I would be happy to answer any questions.
Bueno, sí, bueno, pues una pregunta, yo decía que también que hay preguntas a distancia. Comenzamos por aquí. Ok, ¿alguien tiene alguna pregunta? No, no, I have a question.
Yeah.
Okay, so how does this reconcile with the work of Xiao Chen, where he described this like gustotopic lab of the brain?
Right. And so you talk about the work from Charles Zucker's lab. And so I could talk to you about that for a great deal of time. A lot of what I would say I would want to do one-on-one. But I can tell you that there have been 3 other labs. So what we're doing is electrophysiology. We put our electrodes all over GC. We do not see hotspots. We do not see empty spots, because another big part of that work was that for some reason, there's a great deal of this thalamo-recipient part of GC which magically has no taste responses, which should be a warning. But anyway, we don't see anything like that. But more importantly, we have 3 sets of colleagues who have done exactly what that group has done. Calcium imaging of that region during taste responding, none of them see it. And so the easiest answer I can give you is that those results are unreplicable by any other lab, and so we don't worry about them. But I can tell you offline how those results probably occurred, but it's not a pretty story. But again, I'd even go on and say, nobody else sees these. I got together with a group of 9 big shot taste people to do a review article. And these are 9 people who can't agree on breakfast. These people can't agree on whether it's raining out. When you get these 9 people together, they all agreed on one thing, which is those results are irreparable.
We assume that this processing is like a distributed processing. I saw your guys work like from interesting.
Right. Yep.
That also applies for like.
That's a great question.
That's the case. For instance, when you eat, you feel like happiness. But if you have eaten so much and you're very satiated, then you feel aversive. So what happens?
That's a great set of questions. So the first question is what we see. We mostly did bitter, and the reason was simple experimental simplicity. Those gapes are so easy to see and so easy to effectively measure. We're like, let's just start with them. And in fact, I've got a trio of my people right now. Most of our analysis of gaping is done using EMG, electromyography, because that way the animal can be free to move, and we detect the gape in jaw movements. I have 3 of them working on EMG to try to detect and figure out what exactly is the behavior that determines the intention to swallow. More complicated. We're almost there, but more complicated. And so we'll have the explicit test of that question for you within 12 months. But I can tell you from the electrophysiology that the likely answer is that everything that I've said about bitter also holds for sweet, in that in the electrophysiology, we see a transition in the sweet response that's around the same time. And that the electrophysiological evidence that we show you for the bitter response is something that occurs to all the tastes. And so likely the answer is yes. Don't have the direct test for you yet, but we'll have it next year. The next question was about satiation. And so, yes, and this gets back to something that I've already been talking to Pascal about. We don't want you to think that what we've discovered here is the way the system functions. In my opinion, there's far too much of that going on in neuroscience. This idea that once you understand the circuit, you've got it. The fact is that the brain is a very dynamic place, massive numbers of nonlinear interactions. If you come to my afternoon talk, I'm going to show you how the sense of smell involves one brain region under one circumstance and doesn't even involve that brain region under another. The brain system works differently in different circumstances, and all sorts of things change that. And one of them is satiation. And in fact, we have looked at that. And to send a shout out, over 15 years ago, Ranier Gutierrez, one of his first big papers in taste, done with our friend and colleague, at Duke, showed that satiation changes the way the animal responds. And we see exactly that what we show you lasts as long as the animal is interested in consuming. And that, in fact, one of the reasons we do our experiments the way we do is that we can avoid satiating the animal across an hour by giving it very small doses. But if we up the dose or up the time, the animal becomes satiated, and these things change. We also have data which are also going to be very of great interest to Pascal's lab, but I don't have time to talk about today, which we specifically use conditioned taste aversion to make a taste that was palatable and turn it aversive. And across that change, you specifically change what's going on in that late epoch. That the middle epoch stays mostly the same, and after the transition, it's a different response. We'd like to say, and Pascal was complaining about how people say this, that the response to what was sweet now looks like the response to something that's bitter. That's not really true, but it does change. It does change drastically. And so that is a very good point, that as you adjust exactly what the animal is doing and exactly where the animal is, you change what happens here. I'll even go so far, you get way more, you asked several questions, so you get lots of answer. My very first postdoc, Alfredo Fontanini, in his own work at Stony Brook, has moved on to looking at what happens when the animal knows that the taste is coming. Another thing that we do very carefully in our work is the animal is passive. It's sitting there and all of a sudden gets a taste in its mouth, which is, by the way, ridiculously ecologically unsound. Alfredo compares that condition to when the animal knows that the taste is coming and is getting its own taste. And the epochs that I showed you are still there in Alfredo's data, but they get squished toward the beginning. They get squished earlier. And so just like we were talking about, when the animal, even if it doesn't know what taste is coming, when it knows that a taste is coming, the dynamics happen quickly. And so what we've seen is the way the dynamics work in the specific kinds of experiments that we show. You can monkey with those in a number of ways.
Depend on gender?
Great question. No. The simple answer is no. We see this in both female and male rats. But as you probably know, as someone who asked this question, the estrus cycle of the rat and the mouse is 4 days long, which, by the way, you should be grateful that you are not a rat. And in the work that we've done in the past, we've ignored that. And so we've gotten females across the estrus cycle averaged together. And in that average, we see essentially the same thing we see in the males. We've only recently smartened up and started actually asking, if we very specifically keep track of the estrus cycle, what happens? And so far, we've only got a behavioral study on that, where we, well, that's not true, a behavior and pharmacology, where we used vaginal cytology to capture what phase of the estrous cycle the rat was in, and asked what the animal was consuming. And what we see are fairly robust differences from phase of the cycle to phase of the cycle. And so we haven't done electrophysiology yet, but I would suspect that ultimately the answer to your question is going to be sometimes the female is like the male and sometimes it is. I can't tell you more than that. All I can tell you is it really was lovely, because we did a whole literature search, and it turns out that the general belief that there's a phase of the estrous cycle somewhere near ovulation, when, for instance, chocolate tastes particularly good, was absolutely proven in our data, that just around ovulation time in our female rats, tastes that are ordinarily good are that much better. Tastes that are ordinarily bad are that much worse. Having trouble getting it published. It actually offends some people.
We have a question.
Mm-hmm.
Do you plan on studying how this response affects the decision making or choices made by the animal?
So we consider, I may not be understanding, and please feel free to ask a follow-up if I don't get it right. We consider what we're looking at to be decisions made by the animal. And again, that's getting back to the very beginning. One of the reasons I study taste is because when you put the taste in the mouth, the animal is going to make a decision. That decision is either going to be spit it out or swallow it. But I suspect that the question is about more cognitive style decisions, about what the animal wants to do having received a taste. And we have done a little bit of that. We have done a little bit about what if the animal is in a situation where one taste means one thing and another taste means another thing. I keep looking at you as if you're the person who asked the question. To be completely honest, we've only done one paper on that topic, and it is entirely inadequate. It showed just some basic details that aren't even the most interesting stuff that I got to say. And so we haven't done a lot of that yet. We'd be eager to do it in the future. I haven't had a student who wants to set up, for instance, complicated learning tasks when they can do something so much easier.
Yeah. Go first.
So I'm just curious about something. So you mentioned that after you give quinine to the rat, he's gonna have like an immediate response and he's gonna reject that flavor. But what if you try to condition to something good like right after? Is it possible that you would get like a true response and maybe he would take it longer to reject or even actually like it because he knows that after he's gonna have something good?
Yep, that's a great question. And in fact, we haven't done it with electrophysiology for reasons that will become very obvious. But we've actually got another paradigm that we use in the lab that I will talk about this afternoon, called social transmission of food preference. And the way it works, and I'll explain it in great detail this afternoon, is essentially what one rat likes, the other rat will like. And so if one rat eats something and then the other rat has an interaction with that rat, that second rat will like that, whatever was on that first rat's breath, even if it's a bitter taste. And so, and I've been trying to find this article. There's an old article, probably by the classic ethologist Bennett Galef, that explicitly showed that rats don't gape to a bitter taste if they taste it after having smelled it on the breath of another rat. And this, by the way, for those of you who are keeping score, this puts the final nail in the coffin of any theory that suggests that there's just a hardwired circuit, because in this situation, the animal, the very first time it tastes that bitter, does not reject it. And so that really does happen. We haven't looked at it in any way electrophysiologically for the simple reason that social transmission is an impossible paradigm to pair with electrophysiology. It's hard enough to be able to nail down exactly when to call stimulus onset under ordinary circumstances, even what we do, because there's a delay from the time you send the command to deliver the taste to the taste hitting the tongue. You have to account for that. In this kind of study, you have to account for not only the delay of the odor hitting of the odor on the other rat's breath hitting the animal's nose. You have to account for when that first animal breathes. It's really hard. But it's pretty clear that we can make what was supposed to be a negative taste not negative, and we're pretty confident that if we record on those, we will see a shift in that late epoch that's different from, that's sort of the opposite of CTA.
My questions are follow-up of my colleagues. So first, the follow-up of Isa's great question. What about just giving postprandial rewards like calories? Would it work? Would it be simpler than?
So the main reason we haven't done anything like that in this context is simply that it takes a few days for one of those paradigms to work, and we would most like, we want to record from the same neuron before and after. And another, I spend a lot of my time being snarky and dismissive about a lot of things that neuroscientists, my other neuroscientist friends do. And one of them is insisting that you can put a set of electrodes in the brain and record from the same neuron for 7 days in a row. You really can't. The brain is mushy. The animal just going like this moves the brain, and neurons maximally, the cell bodies are like 20 microns in diameter. And if you really think that your electrodes can keep recording from those same neurons as that brain shifts around across several days, then you're engaged in some fantastical thinking. And so with the sort of paradigm that you're talking about, it just takes too long. But we're working on that because that is a great thing to do. We are doing that sort of paradigm in other studies. And one of the things I'd love to talk to you about is if there is a way that we can make that happen really fast, we would love to do that. But that was only your first question.
Yeah, and I guess I'm not an electrophysiologist, as you pretty well know, and I guess I was involved in some magical thinking because I just read the paper and believe that is pretty.
Yeah.
And that goes to my second question, and it's related to Vito's question. I think many times as scientists, we look for try to simple models because in simplicity sometimes there is beauty. You know, I think we all agree that the 2011 science paper like by Zucker's lab is maybe
simple.
Suspicious, but it's suspicious, but at the same time it's simple. So you are probably familiar with more recent work, maybe try to find a middle road. The Piantadosi paper on the PLA recording. And saw there is obviously there's no identity anatomical segregation. Some later, even Tonagawa work saw segregation, but based on palatability, but much more recent work saw, it's much more intermingled, but there may be some spatial segregation and would make things slightly simpler. What's your take on that?
I could go on for an hour in response to that question. And I'm going to only give you the part of it that isn't combative against individuals. But we have plenty of time. And so let's instead talk about Occam's razor. I agree there is beauty in simplicity. My child, who is now in his teens, he's a simple creature and it's beautiful. But many people, and I say this as someone who has a very strong background in philosophy as well, many people misunderstand Occam's razor to say simpler is better. That's not what Occam's razor says. What Occam's razor says is don't complicate your theory beyond what's necessary. And so that doesn't mean a simple theory works when it's not a simple theory that works. It means that however complex you have to get, don't get more complex than that. And so and this is one of the reasons that in case it wasn't obvious from looking at my CV, I have zero interest in the journal of Science and Nature because you have to simplify your work for them. And that is something that flies in the face of really good scientific rigor, and I will have none of it. And so I don't believe in simplicity for simplicity's sake. I believe in simplicity as much as you can get away with. And let's be honest, the brain cannot possibly be a simple. I do, however, more specifically, the work that I find most compelling in that realm is work from the Swiss lab, work by where the first author was Acola, and that work is spatial coding in GC. I remember, by the way, being a reviewer of this paper, and at that point being asked by my colleague, "Did you hate it? Did you hate it?" And the problem was, I didn't hate it. It is a paper that shows spatial organization, and it is a paper even that shows, we don't have a board. It shows, oh, we do have a board. Do we have a dry erase marker? Okay, so I can just describe it. I can describe it. It shows, and this does end up relating to Chen et al. It shows that there is a huge middle region of gustatory cortex that is multimodal, not non-responsive, multimodal, meaning that it responds to the different tastes. Thank you so much. All right, taste cortex is found at, huh? No one can see that, huh? Okay, let me just return to my interpretive dance. All right, let's try one more. Taste cortex can be found because it lies, there's the rhinal fissure and there is an ascending blood vessel, and taste cortex is around it. It's easy to find in imaging. And what everyone except Chen et al. Have found that in this middle section, there are neurons that respond to everything. And Acola et al. Found that in the middle section, there were neurons that responded to everything, but they also showed, and this was, it was subtle, and I think that it's possible that my other colleagues' technique was not subtle enough to see this. They saw a region up here that was just sweet, and they saw a region back here that was just bitter. Not only that, these regions actually roughly, very roughly, correspond to the hotspots shown by the Zucker group. Now, I have talked at length to these Swiss guys, and what they did not find is that these neurons up here responded more strongly to sweet than the neurons in here. The neurons back here didn't respond more strongly to bitter. There is, however, a bit of a spatial, and this, by the way, is also correlated to pile more evidence against it. There are people who took the receptor work that's been done, and they did put transsynaptic tracer into either taste cells with the bitter receptors or taste cells with the sweet receptors. And what they found is they're all in here with a little bit of a spread out there, and they're all intermingled. So I believe this, and I believe that part of the explanation, although this doesn't get all the way there, because again, these were not the strongest responses to these stimuli. These were just where the edges were. That's part of the reason why the Zucker group saw hotspots, part of it. But, and it is very much the case that there is spatial information there. Furthermore, they went on to show in a paper that was very analogous to the EFIS paper that we did on taste aversion, that when you give an animal conditioned taste aversion, you shift the spatial layout just a little bit. And so I think that stuff is real. And I think to that degree, any full-fledged theory we're going to have is going to have spatiotemporal components to it. And in fact, even if this wasn't true, the fact that you have a temporal response, you have a neuron here that fires most during the first 800 milliseconds and then less. You have a neuron over here that fires less during that first 800 milliseconds and fire more. There is a spatiotemporal pattern that we're not yet dealing with at all. And I think that at that score, I agree with you that we're going to have to eventually take all that into account. But I don't think that's the same thing. And maybe it's because I'm kind of an asshole. But I don't think that that's the same thing as saying, well, everybody's a little bit right.
Why didn't you choose DLA instead of CEA?
That's a great question. I don't know who you are, but So my first choice was CEA. And in fact, the Brian Sadaka, who was the first author in the paper that showed the sharpening of the transition, that was his second paper. His first paper was a sort of a warm-up paper for that, in which he was just showing how these epochs worked in a much more controlled way. And we were like, let's take this opportunity and the fact that you have incredible technique, Brian, and let's also implant the amygdala. Let's implant CEA. And we did. And I don't talk about it, although it's in his first paper, because, and I'm going to be completely honest with you, we couldn't understand what we were seeing in CEA. It doesn't fit in with everything else I've been telling you. We've since then gone on and recorded from the hippocampus, where things also don't fit into what I've been telling you. But now we've come a lot further and we can start making hypotheses about it. We're going to go back to CEA now. And in fact, Jean-Yu Lin, my senior postdoc, that is his goal career-wise, is to bring CEA back into it, because it makes perfect sense to make that an integral part. Our CEA recordings, the taste response in CEA was, we didn't see anything until well after the first half second. So while taste cortex was already pumping away, CEA was still just waking up, and we don't know why, but we're going to find out.
Dr. Kat, thank you again for this.
Thank you, everyone. This was super, a lot of fun for me. Thank you very much. Oh, thank you. It was nice to see you all. It must be obvious that I love to argue. It must be obvious that I love to interact. Please feel free, any of you. I'm easy to find, and Pascal has my email address and my WhatsApp address. Anybody who wants to reach out and ask more questions, anybody who might be coming to the Northeast and wants to see the lab, we are open. Okay? Thank you.
Viendo a los en el departamento, 2 semanas tenemos un nuevo seminario enfocado en la parte económica. Muchas gracias, que pasen una excelente.
Muchas gracias.